The overall goal of this work is to image the nanoscale distribution of phosphatidylinositol 4,5-bisphosphate in the cell plasma membrane using single-molecule fluorescence localization microscopy.
This method helps answer key questions about phosphatidylinositide signaling, such as what's the nanoscale organization of phosphatidylinositides and their signaling complex in cell membrane.
The main advantages of this technique are the high spatial resolution and the bypassing of detergent treatment, which is a major concern in phospholipid morphological studies.
This approach includes 3 major steps. Step 1, the plasma membrane sheet preparation from live cells. Step 2, phosphoinositide labeling and single-molecule fluorescence imaging. Step 3, single-molecule localization and imaging reconstruction. On the day of the experiment, coat cover slips with 1/2 to 1 milliliter of PDL. 1 or 2 hours later, absorb the excess PDL with tissue paper from the edge of the cover slip. Then, store the cover slips on a metal plate at 4 degrees Celsius. Next, wash pre-transfected INS-1 cells that have been grown on cover slips with 1/2 to 1 milliliter of ice-cold PBS containing 1 millimolar EGTA. Repeat this wash 3 times and absorb the excess PBS. Now, using tweezers, place the cover slips with the cultured cells onto the PDL-coated cover slips cell side down. Do this on the chilled metal plate. Then, transfer the plate